Okay, guys. So what we're going to be doing today is we're going to set up a lab that is going to run over a couple of days. Okay. We're just going to, like I say, kind of get it ready. What we're going to be doing here is looking at how a plant transports water. Now, last week we talked about the forces that a plant uses to transport water. What was the main, most important force that moves water up a plant? Osmosis, right? As water evaporates from the leaves, the minerals that were in the water get left behind, and that creates a salt imbalance in the spongy layer of the leaves, and then that causes water to be pulled by osmotic force, okay, up the plant. Of course, the polar nature of water helps in holding the water in the tubes, but doesn't actually provide any pulling action, okay, up. It's osmosis that does that, okay? So that's the problem we're investigating is what forces move water through the plant? Well, we kind of just talked about that, so obviously that is going to have to be part of your hypothesis, okay? Now, identifying our manipulated, responding, and controlled variables. The experiment's going to work like this, okay? You're going to take a cutting of a plant like this. I've got a whole bunch of them in the lab, okay? They're called Dieffenbachia, okay? You might want to write that as part of your observations here. Dieffenbachia. I don't know why they're called that, but anyway, okay, that's this kind of plant. I generally pick it because you can see the dye in the leaves a little bit better because the leaves are light in color, okay? So we're going to be taking a cutting of these and placing it in dyed water in a graduated cylinder, okay? The reason that we're cutting them off, okay, like we're altering the plant here, right? Okay, we're not taking this plant out of the dirt and sticking the whole plant roots and all in the test tube. We're only sticking a little bit of stem and the leaves in the food coloring or the dyed water, right? Everybody follow me there, right? So we're changing the plant a little bit here because we want to eliminate any sources of force other than what? Other than osmosis. And that osmosis or that osmotic pressure is formed in the leaves. So if every other part of the plant is taken off, then if the plant moves water, we know where the force came from. Agreed? Okay. So something about how we're manipulating the plant would probably be your manipulated variable. Okay. We're going to be watching for a couple of things throughout the day, or throughout the experiment, which will be a couple of days. Okay. The things we're watching for, which are usually what variable? Okay. We're going to know how much water is in the graduated cylinder, and we're going to be recording a before and an after, okay, and see if there's any change in the volume. But we're also going to be looking qualitatively at the appearance of the plant to see if we see any dye moving into the leaves, all right? So we're essentially looking at two things here, okay, to indicate whether or not water is being moved. Okay, so those two things will tell us whether water is moving within the plant or not. Now, controlled variables. Controlled variables will be anything that could affect how much or how well a plant can move and transport water. I will let you think up those things. You need 3 of them. Okay? 3 things that we are, at least in some way, keeping the same over essentially a 3-day period. Three. Okay, your hypothesis needs to be an if, and then statement that talks about how plants move water, what you're doing, and how you'll know if the forces you predicted are actually working and have transported water. Okay, I'm kind of weaning you off giving you the whole hypothesis, but those are the things that need to be in there in an if, and then statement. No, you got to restate this in your own words, form of a question, same as always. Okay? You didn't have to do it on the cell transport one because that one was a shortened version, but this one you do. Okay. All right. So I need you to pay super close attention here to how this procedure is going to work, because there's some things that you absolutely have to do right when we go over to the lab, okay, or the lab won't work. Okay, first thing is you want to get a fair chunk of the plant, at least 2 leaves. Okay, at least 2 leaves and a little bit of stem. Okay, let's say we'll go into groups of about 4. That way we're sure we have enough plants to go around, because I have 3 classes that have to do this lab today. Okay, so try and get, like, maybe, if I'm looking at 2 leaves, I would maybe cut right about here. Okay, if I was looking at these top 2, okay, maybe down here, something like that. Okay, they don't necessarily have to be connected. You could get kind of 2 separate leaves if it was kind of picked through by the time you got there, right? But you need at least 2 leaves, okay, in order for this to work, and you need enough stem that it would be able to reach down into the graduated cylinder far enough to be in contact with the water for at least 2 days. Okay, a lot of people are like, oh yeah, it's in the water, and it's in the water like 2 millimeters, and then the next day when 2 millimeters of water have evaporated, their plant isn't in the water anymore. Okay, so you need to have enough stem, okay, that it can fit down into the dyed water. Okay, and I'm going to give you some more instructions on how to do that, but you can see here in this diagram, okay, that it is quite a ways down. This is where the bottom of the stem is. All right, so it's quite a ways down into the dyed water. Everybody follow there? Okay. All right. Now, the problem with using the graduated cylinders is that the graduations, that is the markings and measurements, don't go all the way to the top. Okay, so we are going to use 2 different cylinders. We're going to mix the colored water in a 100 milliliter graduated cylinder. Those are the big ones, the one you see in the picture here. The lab is actually going to run in a 25 milliliter graduated cylinder. Okay, now we want to know how much water we start with, but we want to fill this pretty close to the top. Obviously not all the way to the top, because if we fill it all the way to the top and then put the plant in, the water will come out, and then we won't know how much we've lost. Okay, so we want to fill before we put the plant in, let's say to maybe about here or so, maybe a little higher than that, okay, and then put the plant in. But the problem is the graduations, the measurements, only go to about here, and I won't know how much water is in here if I mix everything in the small cylinder. So you're going to mix 100 mils of water in the big cylinder, exactly 100 mils. Then you're going to pour from that cylinder into the small one until it's about right. So it's about here. Then you'll look at how much is left in the big cylinder. Okay, 100 minus however much is left is your starting volume of water. Almost nobody wrote that down. So I'm going to say it one more time so that you write it down and don't screw it up. I think it's in the procedure in the PDF that I gave you, but some of you were kind of zoned out there. Okay, you're going to mix 100 mils of water in the big graduated cylinder. Then you are going to pour water from the 100 mil cylinder into the 25 mil cylinder until it's close to the top. And then you will look at how much water is left in the 100 mil cylinder. 100 minus however much is left is how much is in the small cylinder. It will be somewhere around 32 milliliters or something like that. It's usually about how much they hold if they got the right amount in them. No, I'm just letting you know. Okay, that's how you're going to make sure you got the right amount in there. Okay. When you are preparing the dyed water, okay, 3 drops of food coloring is lots. Okay, any more than that and it actually starts to get kind of like thick and soupy, and we don't want that. Okay, what colors should you use? Not green, yeah, or yellow for that matter. In fact, I've just stopped putting them out. It was funny the first few times, when people did that, and then 2 days later, the scooters didn't move anything. Yeah, yeah, it sure didn't. Okay, yeah, it ceased to be funny and just became sad. So I don't put the yellow or green out anymore. So red or blue, or you can mix them and get purple. Sweet, you guys remember your grade 2 science. Good. Okay. All right, so we'll look at that. We'll have those as our colors. Now, once we have in getting the graduated cylinder ready, once you put the water in, now we want to make sure we don't lose very much water to evaporation, not through the plant. And if we just stick a plant in a graduated cylinder, there's a lot of open space that water can just evaporate straight out of the cylinder from. So we've got to create some sort of seal as best we can over the top of the cylinder. So once you have prepared the graduated cylinder with water, you'll come up to the front of the classroom and I'll give you the finger from a rubber glove. Okay? Okay? Yeah, some of you got it. Some of you are a little slow. Okay. Okay, so I'll just cut some fingers off of the rubber gloves, and we'll just slide those over the top of the graduated cylinder. They fit pretty good. They make kind of a nice tight seal. Then you just need to poke a small hole, okay, like with the tip of a sharp pencil or something like that. Okay, poke a small hole in the glove or in the finger, okay, and then push your piece of plant through that hole. Don't make it big enough for the plant at first, okay? Let the plant make the hole a little bit bigger, okay? And that way it kind of creates a pretty good seal around it for our purposes anyway. Okay, last thing that's really important is how you prepare the plant. Okay, when you cut the plant, you need to cut it under water. Okay, so I'll have the front sink completely filled with water, okay, and I'll have the plants in their pots just sitting in that water. Okay, so you'll have to take the scissors, okay, hold the plant underwater and cut it off, and then before you pull the plant out of the water, put your thumb over the cut end. Because if you take it out and your thumb isn't over the cut end and you carry it back to your desk and you set it on the table and you get stuff ready, air will get into the xylem. And if air gets into the xylem, it's going to interfere with the polar nature of water, the hydrogen bonds, and your plant will probably not transport any water. Okay, so we need to make sure that the bottom of the plant stays sealed. That's why when you buy roses at the grocery store or any kind of flowers at the flower shop or whatever, they always come with that vial of water on the bottom. Okay, they never have them sitting with the ends exposed because they won't last very long. Okay, so we want to make sure that we have the end covered. Okay, the whole graduated cylinder with the rubber glove and everything is all ready to go when you cut your plant. Okay, so you cut that plant, take it over, and then quickly take your thumb off and put it in. Okay, at that point, I'll give you a piece of tape, put your names on the tape, put it around your graduated cylinder, and the lab is prepped. Okay, then we just wait a couple of days to get our data. I would suggest taking before pictures of your plants. Okay, that way you've got some good comparison pictures of the plant to look at for Wednesday when we go back in there and look again. So take pictures of the top of the leaf and the bottom of the leaf, okay, so that you have some comparisons there, and maybe even take a picture of as much of the stem as you can see as well, okay, in case there's any changes there that you can then talk about in your analysis. Okay, questions on that? Okay, so that's the procedural part. Okay, obviously when we're all done this, we'll have a second set of observations, our after observations, that'll be pictures of the plant, other qualitative observations, as well as the final volume. Of course, the final volume we'll take by just taking whatever's left in the graduated cylinder and pouring it back into the big one and recording the final volume. Okay. For the analysis, okay, question number one is just calculating the difference between the before amount and the after amount of water. That's an easy calculation, just subtract the two, okay? Question number two, what evidence do you have that water followed the path discussed in class? Okay, you can insert, you should insert your pictures that you take into the observations part, but you can certainly talk about them in this question, as well as the change in volume if there is any, okay? And thirdly, predict the effect of adding salt to the water. Describe how you could test this prediction. So tell me what you think would happen if the water was, instead of being just tap water, was salt water, okay? And then kind of describe how you might test that, set up a quick procedure, okay? The quick description of a procedure of how you would do that, okay? And then conclusion, same as always, evaluation, okay? Sources of error and how you could fix them. And then the class discussion, obviously we'll do that in class, so you don't need to worry about that. Okay, questions there? All right.